Okay, so I'm going to look at the cell transport topic today. So we're going to start with the structure of an actual cell membrane. So first thing I'm going to do is simply draw a cell membrane. So the cell membrane is composed of a phospholipid bilayer, like so. Some of those phospholipids have carbohydrate groups sticking out of them. And then interspersed within phospholipid bilayer are some proteins that go all the way through. There are some proteins that either will be found on one side or potentially the other side of the membrane. Some of those proteins will also have carbohydrates groups sticking out, and interspersed between the phospholipid tails is cholesterol. So if we label this, so we know we're clear what we're talking about, this is a phospholipid bilayer. We'll look at more of phospholipids themselves in a minute. Where one of those is a phospholipid. Here we have proteins. Here is another protein. The red thing that I put in between, that's cholesterol. And these are carbohydrate chains. Which makes this a glycolipid. And this, over there, a glycoprotein. Now, if we start by looking at the phospholipids themselves, then we can see that their structure is drawn as a circle with essentially two tails. This is the head. It's made up of the glycerol and the phosphate group, and it is what we call hydrophilic, which means it likes water. Whereas these are the tails. They're made up of fatty acids, and they are hydrophobic, which means that they repel water. Now, because of this is why the membrane is a bilayer, because both the inside, the cytoplasm, and the outside are composed of water. So the heads, which like water, which can cope with the water, they face the outside of the cell and the inside of the cell, whereas the tails are hidden in between. Now, this creates a little bit of a problem because you have this phospholipid bilayer, like so. I'll draw it again down here. Where you have this hydrophobic region in the middle, or sometimes it's called a hydrophobic core. It stops certain things from being able to diffuse through that phospholipid bilayer. So the only substances that are able to actually diffuse through between the tails, like there or like there, they've got to be small, uncharged, lipid soluble, and non-polar. Any other particles, so the large ones, the charged ones, the water-soluble ones, and the polar ones, they can't go through by simple diffusion. They are going to have to go through the membrane by a different route. So, what else was in the membrane? We said already that there were these proteins within the membrane, some of which were on the outside or inside, some of which went all the way through. So if we look at the role of proteins within a cell membrane, we can split it into a number of things. So first of all, they can be transport proteins. Transport proteins fall into one of two types: channel or carrier. And we'll talk more about those in a minute when we look at facilitated diffusion and active transport. They could be enzymes. This is quite common for the proteins that are found just halfway on the inside of the membrane. They might be part of an enzyme chain. They could be receptors. Receptors are proteins that bind to substances coming on the outside of the cell. So for example, insulin binds to a receptor, so it can cause an effect within the liver cell, or the neurotransmitter in the synapse binds to receptors, so they cause an effect within the neuron. They could be for cell recognition, and this is often the case for those glycoproteins. So where you find those carbohydrate chains is normally the outside of the cell, and they're partly used for the cell to recognize that cell as belonging to it. They can also be used in cell adhesion to actually stick the cells together. Okay, there are other functions, but we'll just stick to those for the moment. The other thing that was mentioned in the cell was cholesterol. Cholesterol binds to the fatty acid tails, and it affects the fluidity. So at low temperatures, it will make the membrane more fluid. Okay, now the cell membrane structure, if we go right back up here, it doesn't change wherever in the cell you are, whether you're talking about the plasma membrane on the cell surface, or whether you're talking about membranes surrounding organelles. They all have the same structure. Obviously, the proteins within them may be different depending on their function. So the next thing to think about is, how do substances get across? So we're going to look at a few different processes. The first one to look at is the one we're used to from GCSE, which is diffusion. So diffusion is the movement of a substance from a high concentration to a lower one until equilibrium, which means until the concentrations are equal on both sides. And the only substances, as we said, that can diffuse are small, uncharged, lipid-soluble, and non-polar. So for example, carbon dioxide, oxygen. Okay, they are capable of doing simple diffusion through a plasma membrane. So what about those substances that are going down a concentration gradient, so in other words from high to low concentration, but are charged or large? They do it by facilitated diffusion. So facilitated diffusion is like diffusion. It's the same process. So it's the movement of a substance down a concentration gradient and that's important. We use the word down or against when we're talking about concentration gradients. But this time, we're going through either a channel or carrier protein? Now, what do we mean by a channel or carrier protein? So a channel protein is simply as it suggests. It's a protein which has a shape in the middle, like a tunnel, through which your molecule, your large or charged molecule, goes through. And it has to be the perfect fit. There'll be different channel proteins for different molecules. Think of it a little bit like a shape sorter, a child shape sorter. The right molecule will only fit through the right channel protein. An alternate protein is a carrier protein where the substance binds to one side and then the whole carrier protein changes shape, but it gets dropped off on the other side. It's a passive process because the substance is going down its concentration gradient, but it's worth knowing that either can be used. Okay, what about the next one? So, osmosis. Osmosis is in particular the movement of water. And we describe it as going from what we call a high water potential, symbol for water potential, to a lower water potential through a partially or selectively permeable membrane until equilibrium. So what do we mean by high water potential, low water potential? That's really important. So high water potential is basically as it suggests. It's the willingness of water to move. So if you imagine a solution composed only of water, that's got a lot of water molecules in it. The water potential is what we call 0. The minute you start to add some solutes in there, you're going to lower the water potential. Now, lower than 0 is going to be a negative number. So, for example, it could be minus 100 kilopascals. So water would move from there to there because it's going to move from a higher water potential to a lower one. So one such question might be, let's imagine we gave you 3 cells. That cell has a water potential of minus 200 kilopascals. That one has a water potential of minus 500 kilopascals. That one has a water potential of minus 1,000 kilopascals. Which direction will water move? So water will always move from a higher water potential to a lower one. So it will move from minus 200 to minus 1,000, from minus 200 to minus 500, and from minus 500 to minus 1,000, until they're all equal. So with the osmosis experiment, for example, where you take different tubes and you put them in water with solutes in it, so different concentrations of sugars or salts, and then you calculate the percentage change in mass. Now it's important we do percentage change and not just change, because all your potatoes start at a different mass and you want to be able to compare one to the other. And if you do it against concentration, for example in molar, then you get a graph that looks like this. So at the very low concentrations, what you will find is that water will enter your potato by osmosis because there's a higher water potential outside in the solution, and therefore your potatoes will increase in mass. Whereas when you put your potatoes in a more concentrated solution, there's a higher water potential inside the potato. So therefore, water will leave your potato and go into the solution surrounding it. This point over here, that's called the isotonic point. It's the point at which there is no change in mass because the concentration inside is the same as the concentration outside. It would suggest here it's about 0.15 molar. In order to know what actual water potential that was, I would then need to go to a graph, and they do exist, a graph that has concentration against water potential. And there's, I don't know what the graph looks like exactly, but there's a graph that looks like this, and you'd be able to read off 0.15 and actually work out what the actual water potential was inside the actual potato. Okay, so just to recap, the ones over here where you've put your piece of potato in a very dilute solution, they gain water and therefore they gain mass. Whereas the ones down here where your potato is in a much more concentrated solution, they will lose water and therefore they will lose mass. And obviously the bigger the difference between the inside and the concentration outside, the more the mass lost, because the bigger the concentration gradient, the bigger the water potential gradient. Okay, we then go on to active transport. Now, active transport is the movement of a substance against its concentration gradient. It requires a carrier protein specifically, not a channel protein, and it requires energy from ATP. It is an active process because you are going from a low concentration to a high concentration. Okay, so what do I want to say about this? The important thing is to be able to compare the two. So, for example, to be able to compare diffusion and facilitated diffusion. So, both go down a concentration gradient. Both are passive, but diffusion does not require transport proteins, whereas facilitated diffusion does need transport proteins. Now, you could be asked to compare facilitated diffusion and active transport, in which case you would talk about active transport going against a concentration gradient, whereas facilitated diffusion goes down it. Active transport requires ATP, facilitated diffusion does not. Both of them require transport proteins, but active transport only uses carrier, whereas facilitated diffusion uses carrier or channel proteins. So you could be asked, sort of imagine a third column in that table, to be able to compare the two, all three of them. So if we add a third column over here, and we put active transport, then you would have against a concentration gradient. It's active, i.e. needs energy, ATP. Passive, no ATP needed. That's what we mean. And it requires carrier proteins. So it's quite easy to be able to compare those processes. Now, the last thing I want to think about is some graphs, because they do chuck graphs at you in this topic, and you need to be able to recognize what you're looking at. So a common graph to have, and there are two graphs that I want to pay attention to. So the first graph is where you have time along the bottom, and you have concentration inside the cell, for example, along the side. And you might get a graph that looks like this. And then levels off. And that would be the diffusion graph. Because the reason it levels off is we have reached equilibrium, 50% of it on each side. Or you could get a graph just going to undo that one for a minute. Again. That does that. Seems to be going a little bit faster. Goes a little bit faster potentially than the one just by diffusion. It's because we're doing a bit of diffusion, and it's also doing active transport, so it doesn't reach equilibrium. Okay, that's if it's basically stuck into a solution with a high concentration outside. Bear in mind that it could be presented sometimes in the opposite direction. So you've still got time. And you've still got concentration, but the substance is leaving the cell. So the concentration will drop and then level off. Again, that's diffusion. Or the concentration will decrease all the way down. To zero like that. And that must be active transport. Again, it starts off a little bit faster than diffusion, and it goes beyond the equilibrium, in fact, all the way to zero. So you've managed to take it all the way against the concentration gradient. Just need to be careful about what's written on the side and what's given you in the information that they write. Now, the last graph I want to look at has something different on the x-axis, and this is really important. It's got the concentration on the x-axis. And then it's got rate on the axis over there. So what might happen here? So we're taking a cell and we're putting it in a solution, and we're measuring how quickly the substance is taken up. And we're doing that at lots of different concentrations and seeing what happens. And you'll get a slightly different graph in. It's really important that you pay attention to what's going on. So you will again get two graphs. One will look like that, and one will just keep going. Just going to draw it again so it's okay, it keeps moving it slightly, but it keeps going. Now, it's very tempting to get tripped up on this graph. This graph over here is diffusion. I know this graph is diffusion because as I increase the concentration, the rate of uptake is increasing. In other words, rate is proportional to the concentration gradient. This one over here, this one in blue, that is active transport. And there comes a point at when, even if I put it in a higher concentration, it can't take it up any quicker. Because remember, this is about speed, it's about rate, it's about how quickly it's being taken up, not the quantity. And the reason it can't be taken up any quicker is because active transport requires carrier proteins. And once all those carrier proteins are being used, then the rate can't get any faster. To give you an analogy, if you had 100 doors to a room, okay, and you ask people to leave through doors, okay, and they can leave through whichever door they like, but leave as quickly as possible. If you increase the number of people from 0 to 10 to 20 to 30 to 40, etc., then they're going to leave the room quicker because there's more doors for them to leave. But once you go up to 100 people, each of the doors is now used by one person. If I now have 200 people in the room, they can't leave any quicker than 100 people did because you can only let one person through the door at once. Okay, so it's the same principle. So it's when all the carrier proteins are saturated, and that's why it ends up leveling off or plateauing. And remember, when you're asked about graphs, look really carefully what is on the side, on the x-axis, what is on the y-axis. Think carefully as I change the independent variable, what's happening to the dependent one, and try and work out what's happening. Always describe graphs via the independent variable. As the concentration goes from 1 to 3, rate increases, then it levels off from 3 to 5, for example, because, okay, look at questions carefully, whether they describe and explain, or just to describe, or just to explain. Describe says, is say what you see. Literally imagine describing the graph to someone who cannot see it themselves. Could they sketch it so it looked the right shape? Explanation is the science behind it, the scientific terminology that's actually required. We will leave it there.